All right. Last time on 113 Questions about Evolution, we talked about the physics, I guess you could say, the embryological development of the flat face trait. Why is it? What happens during embryology that ends up producing the flat face in pug dogs and a variety of other flat-faced dogs? Just a refresher, because it's been a while since I made that video. Dogs that have normal wolf-shaped faces, the long straight snouts, that happens because the top of the snout grows at the same rate as the bottom of the snout. Flat-faced dogs like pugs, the top grows really slow and the bottom grows really fast, which causes them to curl upward. And then in dogs like the bull terrier with faces that slope downwards, that occurs because the top grows faster than the bottom. So that's what we learned last time. And we used the frosting examples, right, to kind of get a feel for how that works at a physical level when the embryo is developing, why it is that those weird shapes end up occurring. But today we are going to talk about the genes that encode for those changes in physical form between different dog breeds. Which mutations in which genes cause flat faces in pugs and the droopy faces in bull terriers. 113 Questions about Evolution with John Perry. Evolutionary question number 14. What specific mutations give pugs their flat noses? The development of plants and animals from embryo to full adult is an extremely, extremely complicated process, and scientists are trying to figure out how it is that different genes and different mutations in those genes give rise to phenotype, so the actual shape of an adult. We're actually not very good at being able to figure this out. It's so complicated. There are so many different chemical reactions involved, so many different signaling pathways that are involved, genes and the environment, they play off of each other. It's an absolute mess to try and figure out how this stuff works. But scientists are doing it, and they're doing so in several different ways. One way is that you can actually turn genes off and learn what it was that those genes were doing before you turn them off by seeing how an embryo breaks when it's trying to develop. And we do that a lot with zebrafish and with mice and so on. And the molecule that's used for that is normally morpholinos, stretches of artificial RNA that can actually turn genes off. If you know the sequence of a gene, you can order a morpholino with the anti-sequence to that gene, and it will silence that gene. And then another way to figure out what different mutations do, how they affect an organism, is to just look at a bunch of different organisms that have different traits, close relatives that have slightly different traits or dramatically different traits. And then you look at their genomes, you sequence their genomes, you look for differences, and you correlate those differences in genotype with differences in phenotype. Again, genotype is the sequence of DNA or RNA if it's like a virus or something. Genotype is the sequence of DNA that an organism has. The phenotype is the structure of an organism once it's developed. The really cool thing about dogs is that there are so many different types of breeds and they have so many different traits that you can actually just study different dog breeds, compare their genomes with the genomes of other dog breeds, and you can start to get a picture of what mutations are causing their unique traits. And to do this properly and to really figure out the actual causation instead of just some random correlation. Correlation isn't always causation, right? To do this well, you need to look at lots of different dog breeds. Try and follow single traits. There are lots of dogs that have flat faces. There's pugs, of course, but there's also Boston Terriers and French Bulldogs. The skull that I have, this actually comes from a French Bulldog. And yeah, very flat face. And then also, there's like the apple-headed Chihuahua also has a flat face. I've actually got one of their skulls around here somewhere. This right here, that is an apple-headed Chihuahua with the flat face trait bred into it. And just look at this. So here's a wolf. Here's a Chihuahua for comparison. Chihuahuas evolved from wolves. So that is insane. That is just crazy that this came from this. Sure enough, it did. So, over the years, lots of scientists have worked on these comparative anatomy studies along with these comparative genetic studies, and they've really done a good job teasing out the actual mutations that are causing these differences in phenotype. And there's a lot of work to be done still, but we've got an amazing start just through comparative anatomy and comparative genomics. One of the genes that we've found that's really important for the shape of the snout is one called RUNX2. And we found that within this gene, the RUNX2 gene, there are two sections that consist of tandem repeats. These are sections of DNA that just repeat in sequence over and over and over again. One of them is labeled Q, the other is labeled A, and these labels were chosen because of the specific amino acids that these tandem repeats code for. So this is a protein coding gene that has two different tandem repeats within it. Tandem repeats are really interesting chunks of DNA because they can go through what are called slipped strand mispairing. This is a very special type of mutation where the enzymes that copy DNA when a cell is reproducing or when sperm and egg are being produced, those enzymes, they'll attach to this stretch of DNA, they'll start trying to make a copy of it, and they can slip, they can get bumped off in that process. And when they reattach, they can reattach in the wrong spot. I'm kind of oversimplifying things a bit here, but what ends up happening is sometimes they can copy a chunk that's already been copied and they make a strand of DNA that's too long. In other cases, a slipped strand mispairing can cause a chunk of DNA to be skipped when being copied, so it can make the gene shorter than normal. These types of mutations are extremely common when you have a stretch of DNA with tandem repeats in its sequence. What scientists found in this 2004 paper called Molecular Origins of Rapid and Continuous Morphological Evolution, what they found is that any mutation that increases the length of Q, segment Q in this gene, relative to segment A, this causes the top of the snout to grow faster than normal. In other words, this is exactly what we see in the bull terrier because the top of the snout is growing faster than the bottom of the snout. And check this out. They actually found that bull terriers have an extremely long Q compared to A segment in this gene, in the RUNX2 gene. And they went kind of back in time. If you look at photos of old bull terriers, if you go back 100 years or so, you'll see that they still have the droopy face, but it's not nearly as extreme. And they actually found tissue preserved from one of these old dogs with kind of the intermediate version of this trait. And they sequenced its genome, and sure enough, there was an intermediate version of this gene here, the RUNX2 gene. The Q was slightly longer than A, but not extremely longer, as we find in modern bull terriers. And this was a really cool confirmation of this finding that, yeah, this mutation, the lengthening of segment Q compared to segment A, really does seem to be what's causing the fast growth on the top of the snout versus the slow growth on the bottom of the snout. Now, as you might expect, the reverse is also true. If mutations lengthen the A segment in relation to the Q segment, the top of the snout will grow slower than the bottom of the snout, and as a result, the face will curl upwards. Now, this mutation alone cannot explain the extremely flat noses that we find in pugs. You actually need more mutations in order for that to happen. But it can explain the kind of, mildly flat faces and upward pointing faces that we find in like Great Danes. So these two stretches of DNA within the RUNX2 gene, they kind of act like little dials. Different mutations over time can dial the face to be stubbier and point more upward, or dial it to be longer and point more downward. And it's a really cool control mechanism that happens here. And these mutations, they happen very easily. And we've seen, again, with the bull terrier, that these mutations are happening still today. This isn't something that, happened a long time ago in wolves and selective breeders just pulled it out of the original wolf population. These mutations are still happening now, and they're still changing the shapes of dog faces. Mutations like this are just a continual source of novelty that evolution can play with, or in the case of dog breeding, dog breeders can play with. A second paper that was published in 2012, they found that there are 5 more stretches of DNA within the dog genome that appear to influence the length and shape of the snout. There are probably lots more, but we know of 5 of them. They were able to pinpoint one of them really specifically. They discovered a single point mutation in the gene called BMP3, and this point mutation dramatically flattens the face. It's found in pretty much all of the small breed dogs that have flat faces. It's not found in the larger dogs like the boxers that have moderately flat faces, but it is found in things like Boston Terriers and French Bulldogs and, wait for it, the Pug. So now we have two independent mutations that both affect the flatness of the nose, and if you add those two together, you get more extreme flatness. If you have just one, you get fairly mild flatness. But then another paper in 2017 found yet another really important mutation. In our genomes and in the genomes of dogs, there is a gene called SMOC2, SMOC2. In flat-faced dogs, this gene contains a large insertion. A retrotransposon has been inserted into an intron in this gene. So an intron is a part of a gene that's not normally coded into protein. It's just sitting there in the middle. And a retrotransposon has been inserted in there. Retrotransposons, for those who don't know, are chunks of DNA that are able to make copies of themselves and do sort of a copy-paste mechanism throughout the genome of an organism. So they code for a protein that then makes a copy of that gene and inserts it into other chunks of an organism's genome. And a lot of these come from viruses. So this retrotransposon has been inserted into the middle of an intron in the middle of the SMOC2 gene. Several really careful experiments showed that this insertion slows the expression of the SMOC2 gene. It makes it so that during embryological development, less of the protein coded for by this gene is produced. And as an end result, you get really flat faces. This mutation alone was responsible for more flatness in the face than any other mutation we found so far, if it is the only mutation that's inherited. And this we were able to figure out by studying different crossbreeds. If a crossbreed has this mutation and not the other two mutations, it still has a pretty dang flat face. It's not as flat as if it has all three of these things. These mutations work in kind of an additive manner, but with this mutation alone, they have a very, very flat face. So let me just kind of put this all into perspective here. Scientists have discovered, there are three different types of mutations that can cause a flat nose to occur in dogs. We've got a slippage mutation, we've got a point mutation, and we have an insertion mutation. These three very, very different types of mutations, completely different origins, they all work together to cause this flat face trait in pugs. You might think that that's not cool. For those who know a lot about these flat-faced dogs, they have a lot of health problems because of these mutations. That said, there is something about flat-faced dogs that really seems to trigger people's parental instincts. Doesn't do this for all people, but for some people, they melt when they see one of these flat-faced dogs. I just find this absolutely fascinating. The messy way in which new traits arise. Next question.